it's a really prominent feature of them. So that's really interesting. You know, it's really part of their biology.
Mm-hmm. But yeah, I wouldn't know which ones they are. They look like little, if I don't know if you can zoom in on that picture there on the
we can try to do that.
The one above this one, yeah. They were.
This one?
Yeah. And they look more like little discs, you know. They're really funny looking. They're not really cups.
Mm. Yeah, I mean, again, if you had his book, I think you could picture match it. I've never spent any time with that book to know what the key is, how he keys them out. And you know, we had LAMS was selling that book for a while, and I kept thinking I was going to get one, and then they all sold out. But you can still get it on Amazon.
Oh yeah, don't shop.
So you might want to have that, Andre. If it's those are the mushrooms you're going to be seeing. I think.
Well, let me suggest a Libris or a Libris instead of Amazon. Don't give Mr. Bozo any more money. He already has too much.
Sorry, but I mean that's what people benchmark it to. That's what they want to know when they're buying it from LAMS, is like how much it compares. And LAMS actually was the winner for a while, and I think that's why our copy sold.
Oh yeah, okay.
But they're gone now. You can, if Rich is still on, he could probably confirm that.
Well, look on Alibris. You could find it used and much cheaper than on Amazon also.
Yeah, and I use Add-All. Do you know Add-All?
Nope.
Add-All uses, here, I'll just show it to you real quick if I'm still sharing. This is how I find books, because you can find super rare books on Add-All that you can't even find on a Libris. So it's www.
Oh yeah. ADDALL.
Yeah. And so it searches a Libris and like 30 other, you know, if you know Abe Books, for example.
Yeah, I think
Abe Libris, it searches Abe Books. It includes Amazon. You don't have to buy there.
Mm-hmm. But Abe is also Amazon, as far as I know.
It got bought?
I think so. Yeah, you really have to do your, research to not.
Sorry to hear that. I didn't know that.
Yeah.
All right. I've lost.
Yeah.
I've lost my. Where are you? There you are. No, that's not you either. I've lost the.
Nice.
I've lost the meeting.
Oh, there it is.
Is that it?
Yeah. Well, that's the first cup, but yeah, maybe not.
I guess I was in PowerPoint. That's why.
Oh, I see.
Hold on. But I think we're done with the PowerPoint. Yeah, so now we're just about ready to start the main feature. So I'm going to end the slide share here. Thank you, everyone. Really appreciate all those contributions, and I'm glad people are still finding fungi. It's been a difficult spring, a disappointing spring. You there, Rich? Thanks again, Andre. I see Danielle. Hi.
Yes, I'm here.
So maybe give it a minute. I have a couple of announcements before I introduce tonight's speaker. Just want to remind people about the call for nominations and also our special meeting. But let's just give people a minute. I don't know if people are coming on. If they're like me, they're a little disappointed with the lack of rain and the sort of excitement around fungi.
Well, we've got 37 people right now, so it's a decent crowd.
Okay. Well, you know what? Maybe if people come in late, maybe I'll even plug LAMS activities again at the end, and I won't hold us up too much right now. So the things I just want to remind people of are that we have a call for nominations for the LAMS Council. It's open to everyone that would want to be a LAMS Council member. This is as prescribed by the bylaws of the group, and these are the positions that are open. I've actually want to point out that we've had the call. It's been out for a couple of weeks, and we're closing it soon. So if you know anyone, I would love to hear from you. And I would especially like to point out that we don't have anyone that I know of so far that's running for secretary. And we need a secretary desperately. I don't know if you know how important the secretary is for the functioning of the the club, but you can't have a meeting without a secretary. Because the secretary records the minutes, and the minutes are part of, they record the votes of the council members, and that's the official record. So it's a really important job. I know it has this name that doesn't sound necessarily important, but it's a really important position. And so if there's anyone out there that would consider running for secretary.
Hey, David,
Yes.
Aaron Thompson just posted in the chat. He would run for secretary.
Wow, thank you. Thank you so much. It's funny when I'm in this view, I'm gonna keep sharing, but thank you. That is so excellent. And if you want to chat about what that's all about, the other thing I'll point out is we did have a little description. Just see if I can get to the next page. A little description, and here's the secretary. So it's couched in this, maybe not important sounding, but it's so important. So I really appreciate your willingness to volunteer to run. And Rich, I'd like you to record that, because we will be putting together the ballot soon. I'm not seeing the chat myself right now. I don't have that in my view.
Okay, well, by recording the meeting, it saves the chat as part of the recording as a separate file, so I'll get everything.
Okay, excellent, thank you. And I'll go back in once the speaker starts. So the other thing I want to point out before we get started tonight is that we have a special meeting, and that happens tomorrow. And what the special meeting does is it changes our election procedures. And unlike some states in the US that you may have heard about, we are expanding the ability of our members to vote by including electronic voting. So with the pandemic, getting to a post office, getting stamps, just even doing snail mail anyway is, going the way of the dinosaur. And so it was actually our treasurer, Rich Collier, who suggested that we do this. It was brought up at a LAMS council meeting, and the council voted in favor of having a special meeting to change the bylaws. We cannot just change the bylaws. The bylaws require either a special meeting or to be brought as an issue at the annual meeting. Everyone present, and that includes any LAMS member, may be present at a special meeting and vote. The voting, it requires a 2/3 majority to pass. So we'll be doing this starting Tuesday at 7 p.m. And I believe Rich has already sent out the link. So if you're a LAMS member, you can log on. You can hear a brief presentation of the idea of having electronic voting that allows anyone to vote from as a member, as a LAMS member, to be able to vote online rather than having to go to someplace in person or send in a paper ballot. So thank you, Rich. I think that's going to be well appreciated, and we'll find out tomorrow. So I'm going to stop sharing, and I'll just go ahead and introduce tonight's speaker, if we're ready for that. So tonight's speaker is Danielle Stevenson from D.I.Y Fungi. And D.I.Y is involved in a number of areas related to fungi. And that includes general education, it includes mushroom growing, it includes bioremediation. And I know there's a lot of interest in LAMS members and among LAMS members in those areas. So Danielle is currently a PhD candidate student at UC Riverside, and she's studying environmental toxicology. And tonight she's going to talk on the topic that I love, the great title, One Person's Trash is a Mushroom's Treasure. And I won't give away what she's going to talk about, but I'd like to turn it over to Danielle. So Danielle, thank you for coming tonight. We appreciate it.
Hi, everybody. It's wonderful to be here. I'm actually a LAMS member, so it's really, it's a pleasure to be here with you all tonight talking about something I'm really passionate about. I've been involved with fungi in lots of different ways for over 10 years and study them now, although this is, what I'm presenting on tonight is more of a passion, an area of passion and interest for me. So yeah, thank you, Kate. So I'm going to go ahead and share the presentation that I made, and I just wanted to invite you. I mean, it's a sort of a casual talk, and so it's geared towards there's aspects of mushroom cultivation and aspects of remediation in the talk, and so I'm going to try to clarify and define what I mean, as I may be using terms that are unfamiliar, but don't hesitate to just call out if you have a question. And you can also pop that in the chat. I can see it kind of pop up when there's a comment in the chat. So first of all, can people see and hear okay?
Yes.
Okay, perfect. Thank you. So there was a lot of introduction to D.I.Y fungi, stands for do-it-yourself fungi, but I'll just mention I actually started D.I.Y fungi in the Pacific Northwest, a very different climate for mushrooms. I was offering mushroom cultures, also known as spawn, and doing a lot of education around how to grow mushrooms for food and medicine from home to farm to community scales, as well as doing research into applications for fungi and remediation of contaminated sites and also waste. So that led me into my PhD, where I now study particular type of fungus called arbuscular mycorrhizal fungi that you may have heard about. There's been some fantastic presentations by Tom Bruns and others that have talked about these special fungi. I'm not going to talk about them tonight. We're going to focus mostly on the decomposers, as we'll get into. But I think that I'm so excited about this potential to apply mycology to address some of the major problems that we're facing in the Anthropocene, which is defined as our current geological age, a period in which human activity is the dominant influence on climate and environment. So you may know we need to feed a growing population and especially provide environmentally sustainable protein options. Food insecurity is rising globally in the US and in California, where 1 in 5 people don't have access to the food they need to be healthy. Environmental pollution is widespread in soil, air, and water. Our landfills are filling up and trash is being shipped to distant places. So for me, this seems like a lost resource, and maybe there's something helpful about that adage, waste not, want not. And maybe there's something helpful to conceive of waste, as Danielle Freitag does, as a natural resource in the wrong place. He says, change the context and you have usable products. So I like to nerd out about what's actually in our waste stream. So here's some data on what is in California's waste stream. And so you may not be surprised, you may have heard that organic waste, so mostly food waste, but also yard trimmings, comprise a significant piece of the pie. Almost 40% of what goes to the landfill is, food waste and yard trimmings. Inerts and other, about 20%. That includes actually wood and construction waste. Paper is another significant category. You can see almost 20%. That's cardboard and other paper waste. And then plastic, over 10% is single-use plastic products. So I thought it was helpful too to get a little more specific. This table shows the 10 most prevalent types of waste, and you can see number 1, food, number 2, lumber, number 3, paper. We see again in the top 10, textiles, other organic materials, other paper, leaves and grass, cardboard, prunings and trimmings. So I think as people who love mushrooms, probably many of you forage and eat mushrooms, maybe some of you grow mushrooms. Many of you might be thinking, hey, okay, paper, cardboard, wood, organic materials, isn't this just what decomposer fungi eat? And so you're right, and this is exactly what got me all inspired about the potential here. So we'll come back to that and build on it. But I need to mention as well that there's unfortunately a whole range of pollutants that actually don't even make it to the landfill. So we have lots of motor oil that leaks from leaky cars, 60 million gallons a year just in California. Kitty litter, 2.5 million tons of waste per year in the US. Cigarette butts are the most commonly littered item. 40% of all lubricant oils wash off into the environment. 50% of homes with septic systems are failing. So we'll come back to actually each of these contaminants with some ideas later on. But I just wanted to bring these up because they may seem small. You might think, oh, a cigarette butt, but cumulatively, these actually have a large impact in leading back into one of those problems we started off talking about in terms of environmental pollution. So I see potential to apply mycology to 1, divert non-toxic waste from the landfill as substrates to grow protein-rich mushrooms for food. I see potential to apply mycology to reduce the toxicity and accelerate the decomposition of polluting waste, essentially creating soil out of formerly toxic materials. And then there's potential to replace toxic and environmental costly materials with fungi as well. So, fungi are principal decomposers in ecological systems and play fundamental roles in soil formation, nutrient, and carbon cycling as well. So which fungi are best suited to dealing with waste? Of course, those decomposers, right? These fungi, whether they're, you know, sort of charming, you know, mushroom forming fungi from the Basidiomycota, like oyster mushrooms pictured here growing on a log, or the sort of important you know, common molds like penicillin from the Ascomycota. These decomposers break down dead stuff. They break down organic matter, the remains of what was once alive, using extracellular enzymes. And they do that because that's how they get their food. Endophytic fungi are communities of fungi that live inside the tissues of plants, not just the roots like the mycorrhizae, but all the parts of the plant. And they earn their living by trading a number of benefits, like helping plants photosynthesize better, and protection from disease, and other benefits for room and board from the plant. So these two categories seem to be best suited to helping us deal with these different problems. I'd like to go a little deeper into the different subcategories within the decomposers. So for folks interested in remediation, it is good to know that there's brown rot fungi, which can degrade cellulose, common component, the fibrous part of plants. And then there's the white rot fungi that have enzymes powerful enough to degrade lignin as well, which is what makes trees rigid and hard. It's a really hard thing to break down, but they figured it out. These white rotters tend to be useful in remediation of organic contaminants. Another thing that's helpful to know, though, when we're talking about waste, is the difference between the primary and secondary decomposers. So primary decomposers like oyster mushrooms and turkey tail like to grow on organic material that hasn't been broken down at all. So like a wood. Whereas the secondary decomposers like the agaricus species, the button mushrooms and shaggy manes, they actually like to eat organic matter that's already been broken down a bit by other microbes or by cows. And so these are the ones that grow on compost and manure. And just for those who haven't cultivated mushrooms before, I wanted to kind of start out by defining a little bit of terminology so that we can all move forward together. So I've already used the term substrate, and that refers to any organic material on which mycelium will grow. It's the food for the fungus that you want to grow. Spawn, I also have used that term. It's just another term for a mushroom culture. It's pre-grown mycelium. And that's what you need if you're wanting to cultivate mushrooms to get started. And then I sterilize. So typically we need to do something to kill or weaken the competition on the substrates that we want to grow on. And that's so that the mushroom we want to grow can survive and thrive. So we usually use heat or hot steam or water to do that, to sterilize. That'll become relevant later when I talk about some of the projects I've worked on. So, so diving into this first category, the potential of diverting non-toxic waste from the landfill and using them to grow mushrooms for food. So this can be done at a home scale. Some of you may try this out this week if you get inspired, but it's also done commercially. And so oyster mushrooms are a great, so these are the Pleurotus species. They're a great one to grow because they are aggressive enough and they're not picky eaters. They can be safely grown on everything from spent coffee grounds and tea leaves to various types of agricultural waste. I even know of a commercial mushroom operation that was growing oyster mushrooms on invasive plants pulled from ecological restoration events. So lots of possibilities. It's organic matter, right? They certainly will grow on paper and cardboard waste, wood waste, and even biosolids. Button mushrooms. So globally, oyster mushrooms are actually the second most commonly cultivated mushroom crop, and button mushrooms are the first. These ones, remember, they're secondary decomposers, so they like to grow on composted waste materials. But remember, food waste is about 40% of what goes to the landfill. So a lot of potential there to compost that and use it as a substrate for button mushroom growth. And then there's other edible and medicinal mushrooms that can be grown on sawdust and wood chips made from wood waste. And so there has been a bit of research into this. There are still some more research that I think would be really beneficial. Research questions I see are, to what extent does the substrate affect the nutritional properties of the mushrooms? Are there any contaminants in these like seemingly innocuous waste, like food waste, that we need to think about and mitigate? And, is this a potential to create livelihoods for people? Again, waste is free. I mean, typically, actually, people have to pay to get rid of it. So there's some potential here to create livelihoods and economic well-being for people. This is another example. This is a company called GrowCycle. I think they're in Portland, and they grow oyster mushrooms to sell to restaurants on coffee grounds, as you can see here. I'm just gonna peek in the chat because I see it. Okay. One of my colleagues at the nonprofit organization Co-Renewal that does mycology research did his master's actually on this really neat subject. He looked at using biosolids actually produced from food waste and dairy manure as a substrate, as a co-substrate to grow oyster mushrooms. And so long story short, they found that these biosolids could actually replace soy hulls. I mean, for those who are cultivators, you often will supplement sawdust that you may be growing on. And so it turns out that these biosolids, again, created from food waste diverted from the landfill, as well as dairy manure waste, was a suitable supplement to grow mushrooms for food. There's so much more potential there to explore, but now we'll talk about the next sort of potential for applying mycology to reduce the toxicity and accelerate the decomposition of polluting waste. So I just want to be clear about the different kinds of contaminants that you need to think about in waste. And so we have metals. These are elemental contaminants. They are examples like lead, arsenic, chromium. They're the kind you'd see on the periodic table. They can't be broken down, but they can be moved around, something to think about. Organic contaminants, however, can be transformed and broken down, degraded. And that's because they're manufactured or derived from something that was once alive. So petroleum products, agrochemicals are in this category. They're examples of products that were actually made from something that was once alive. So what's really cool is that organic pollutants can often be degraded by decomposer and other fungi through those same enzymes they use to eat organic matter. So I'm showing you here the structure of cellulose and lignin, the main components of plants, right? And I'm showing you also the chemical structure of two common organic pollutants. One is a pesticide, DDT, and one is a common, another common organic pollutant. And so actually to the fungus, it looks like dinner. It looks pretty similar to what they would, be applying their digestive enzymes to in a log or in a pile of leaf litter. And so this is a little cartoon just showing the fungal hyphae releasing these different enzymes, licases and peroxidases, that are actually breaking apart these, POP stands for persistent organic pollutant. It's actually breaking it down so that it can ingest the carbon and the nutrients that the fungus needs for its life processes. So there's a number of things one can do if you're excited about exploring this realm of working with fungi to decompose toxic materials. One is that you can actually train or acclimatize a fungal species to be more efficient or fast at metabolizing a given pollutant or food source. So on the left, we have one example with metals, actually, and where we have any mushroom cultivators are probably groaning seeing trichoderma. It's the bane for the mushroom cultivator. We don't like to see it come in as a contaminant when we're cultivating mushrooms, but it's actually a really, really effective remediator. And in this example, the trichoderma was had increasing concentrations of arsenic added into its little petri dish, and it actually became adapted over time to be able to tolerate something like 10 times the arsenic that it originally would have been harmful to it. And this is a picture, one of my own pictures from a project I'll talk about later, where I've sort of acclimatizing a Pleurotus species to metabolize a particular bicycle lubricant. And so this is a common practice in the real world of mycoremediation. This is a particularly inspiring example for me. It's work done by Rosa Valdemar in Mexico called Treatment Alternatives for Disposable Diapers. I need to move there we go. So diaper waste is a major source of plastic to the landfill. So each child produces 1 to 2 tons of diaper waste. This picture is really funny. So Rosa took this on and actually worked with Pleurotus austriatus, so the common pearl oyster mushroom, and she basically mixed in these disposable diapers that use disposable diapers with a little bit of corn. It was agricultural waste from corn, and found that the Pleurotus broke down the disposable diapers by 80% in just 2 to 3 months, recycling the plastics within and producing. Initially, they were hoping to be able to grow, harvest oyster mushrooms grown on these materials. But after testing, they determined it was actually safe enough to be the remaining substrate, what's left over after this growth process, was used as a mulch in agriculture or feed for cattle, which is pretty great. That's a pretty good outcome. Yeah, it's so cool. So, and then here's another approach. Instead of training a given fungus to be more able to tolerate a given contaminant or toxic waste, you can actually find fungi that are already adapted to toxic waste or harsh environments by going out to a harsh environment, or even a pile of the waste that you're trying to work on, and actually isolating and culturing fungi that are found growing. And so that you might be surprised, you might not be, I'm not sure who's in the room, but it might be surprising to imagine that there would be fungi growing, for example, on electronic waste. But in fact, there's fungi everywhere. And this is from a study done where actually fungal strains were isolated from an e-waste recycling facility. And these fungi are exceptionally good at sorbing, basically grabbing onto and kind of stripping toxic heavy metals from e-waste. And so actually today there are biotech companies that actually are able to work with fungi to extract precious metals from electronic waste, and then also remediate and address the toxic metals that are not useful, that are not going to be able to be sort of reused. So the basic protocol for that kind of approach, and this is something I do in my research as well, is I'll actually go out to contaminated sites and Superfund sites and harsh environments and take samples and then go and sequence and identify the fungi that are growing, culture them, grow them out for use in, you can do further screening to identify which ones are best for certain applications. And then ultimately with the hope of being able to work with these fungi as kind of superstars for remediation. So this protocol is from a study by Thomas et al. And it just goes through the basic protocol. It's something that probably many of you have the skills to do, right? So you're collecting fungi from the local environment, you're isolating them to get a pure culture, and then essentially, transferring to a growth substrate to increase the volume of the fungal material, the fungal mycelium, which you're going to typically need a lot of for remediation. This kind of approach was taken by students at Yale who actually identified an endophytic fungus, Pastaloteopsis microspore, which can eat polyurethane plastic as its only food source in the absence of oxygen. So that's kind of exciting. I keep waiting to see what will come from this, because of course, we've got a lot of plastic sitting at the bottom of landfills without oxygen. So it would be fantastic to see. In plastics, as you saw, a big contributor to the waste stream in California, but globally, it's a major problem. And then I'll say too, that actually some molds and yeasts, also from the fungal kingdom, are able to degrade plastics. So actually certain Fusarium and Candida species, but also Aspergillus and Penicillium. So these really common molds and yeasts can also degrade plastics when you basically do a hot composting process with these fungi. There's also potential to work with fungi to filter animal agricultural industry wastewater. So this is another major source of environmental pollution and waste. This is this really neat study done up in Washington, again by Thomas et al. It was a field demonstration of microremediation to remove fecal coliform bacteria and nutrients. So basically, as you can see in this schematic, they created a rain garden with native plants that were inoculated with mycorrhizal fungi. And then they mulched pearl oyster, phoenix oyster, and king stropharia. So these are these charismatic, edible decomposer fungi. And they sort of grew them on alder wood chips in this rain garden and found that it together, there was a 90% reduction of fecal coliforms and excess nutrients from completely untreated dairy lagoon waste over a 17-day period. Pretty good, pretty impressive. So this is something, setups like this I have also tried with leaking septic tanks, with different types of composting toilet systems and other systems where there's there's a concern around bacterial pathogens or wastewater. And yeah, it's pretty effective and pretty straightforward to implement. So a model I've been working on for dealing with common toxic waste is something I call mycodigesters. And I'm really grateful to some fantastic mycologists. Peter McCoy, I know, has spoken at the club. I was inspired years ago. He put out a video or a picture or something of oyster mushrooms in grain jars with cigarette butts in there. This was followed up by Alex Dorr, another mycologist who did a bit of testing on how that worked. Jay Schindler of Fungi for the People, a brilliant mycologist. I took a course with him many years ago, and he showed us that he was playing around with elm oyster, Hypsozygus almarius, to digest bike rags. And Trad Cotter, you may know, fantastic mycologist with the book Organic Mushroom Farming and Mycoremediation, has some really cool ideas in that book about digesting pet waste with king stropharia and oyster mushrooms. So I've definitely drawn inspiration from these mycologists. So I actually worked on mycodigestion of Teflon-based bicycle lubricant soaked rags. And so my basic, well, I'll keep it short, but just Teflon is kind of a forever chemical. It and like I said, when I looked into it, I learned that 40% or so of lubricants just wash right off, and then they persist forever in our watersheds. So I ride my bike, and I went to my community bike shop one day, and I was getting my bike fixed, and I just asked them, like, hey, what do you do with the rags? And I remember the guy just turned red in the face and took me out back and showed me like a mountain of garbage bags full of these rags. So they just sent them to the landfill. So I said, well, can I set something up here? So basically I did the training approach. I worked with Pleurotus because I knew that it was pretty good at dealing with hydrocarbon based substances. So I did the training approach, basically introduced a little bit of it to a culture, and then observed it getting faster and faster at being able to completely consume it. And then I started scaling up. So I started in 5-gallon buckets, and I scaled up to 55-gallon containers over time. So I'd be happy to share more about this project, but it builds into my my overall model, which I'll share at the end. I also followed up, actually was invited to do a study into the cigarette mycodigester option And so I designed this study and undertook it to assess, like people had shown that oyster mushrooms could seem to digest, but I wanted to test it and see, well, to what extent does it degrade the main contaminant of concern in cigarette butts are called PAHs, polycyclic aromatic hydrocarbons. They're also what's found in like car exhaust. Kind of nasty. But I want to see actually like to test like, well, are these pHs getting broken down by the oyster mushroom? How like does effectiveness is it affected by how sterile it is, or like what method you use to inoculate? And so how I wanted to answer those questions, thinking to scale, like, can this be something that can be scaled up? So I actually experimented with, I had a control. So these were just cigarette butts with no treatment of the oyster mushroom. I did sterilized butts inoculated with liquid culture. I did sterilized butts on top of pre-grown mycelium, as you can see here. And then I did unsterilized butts with inoculated with liquid culture and unsterilized butts in the jar of grain spawn. And so this is what I found. So I'll just walk you through it. The control, and this is showing concentrations of these different, there's many different PAHs. And so the ones tested are listed along the bottom. And then on this vertical axes, this is the concentration of each of these contaminants. And so what you can see is that for the most part, I mean, there was a pretty stark difference between the controls. So with no treatment, there wasn't degradation of these contaminants. In general, let me see. So the sterilization did seem to help. I was hoping it didn't matter, but it did seem to help. You can see that the unsterilized butts inoculated with liquid culture did worse comparatively to the others, even the unsterilized butts that were added in on top of fully grown mycelium. But overall, in just a 2-week period, so 14 days was the length of the experiment, there was pretty significant degradation, particularly of the the more simple PAHs, the ones with fewer rings in their structure. But overall, it was, I wish I would have included those calculations, but I think it was something between 70 to 97% effective with any treatment with this pearl oyster. So based on these different experiments, I kind of tried to distill some best practices and things to consider for working with fungi to turn toxic waste into soil. First of all, you'll need to build relationships to collect and contain waste. I mean, it's always helpful to start off with a waste that you produce, like a household waste. That's easiest, but you can also scale up and actually partner with municipalities or businesses to collaborate on collecting and containing this waste. You're going to need to ensure that the fungus has what it needs to grow. And so this is where your mushroom cultivation skills will come in handy. So if you if you're not sure, I'm going to go through the basic things that fungi need to grow in the next slide. A third best practice is that mimicking nature works best. So you can follow the principles of ecological succession, where we typically have primary decomposer fungi like the oyster mushroom that start off, remember, they like to eat something that hasn't already been broken down. So they can actually, through their enzymes, break apart really complex and they can break about really complex, hard to break down things, including organic contaminants and different organic materials into bits that then secondary decomposer fungi, as well as bacteria and other soil organisms like worms, can finally get at. And they continue the process of physical and chemical breakdown. And then typically at that point, you have soil and plants can come in. If there's metals, you want to be careful that you're not planting food crops that might take up those metals. But there's plants that are actually really great at accumulating toxic metals and storing them in their bodies. So the fourth best practice and thing to consider is that when you're working with fungi for remediation, you need to treat, it's not to grow food. There's plenty of waste that is non-toxic to grow mushrooms for food on, right? We got coffee grounds for that and paper and, agricultural waste. But when you're working with fungi for remediation, you're going to need to have a plan for how you'll contain and dispose of any mushrooms that may form. And so again, with remediation, we're working actually with the living organism, the fungal mycelium, the filamentous, threads of mycelium that put out those enzymes to break things down. But if mushrooms do form, you'll have to contain them and treat them, have a plan for what to do with them. So I'm just going to pause because I see the chat going off. Okay, there's a couple questions. I love the exclamation marks. Thank you, guys. That's great. So could some of the digested substrates incorporate the mushrooms? Yeah, so that is exactly what I'll do. I mean, it's good to test to see how toxic, or if there's even any toxicity in the mushrooms themselves. There may not be, really only if there's metals. We expect metals to be accumulated in the mushroom fruit body. I have tested that before. Sometimes, yeah, metals do get taken up into the mushroom fruit body. If the concentration is really high, I just have some kind of sealed bucket. I'll just put them all in there, and you can actually take them. Usually landfills have a hazardous waste disposal part, and you can dispose of them there. That's if you're doing a big project and the mushrooms have, a lot of metals in them, basically. Otherwise, for organic, if they're growing on, so with the bicycle rags, that's not a great example because there were metals. The problem is that there often are metals mixed into things like lubricants and all sorts of products. If there aren't metals, then I wouldn't expect there to actually be, for instance, say pesticides. If there weren't metals in the pesticide and you worked with a fungal decomposer to break them down, I wouldn't necessarily expect there to be, pesticides in the mushroom fruit body that forms, but it's good to test. But that would be an example where you could actually just put the mushrooms, let them just decompose within that system. That's what I usually do. Okay. And so just for folks who maybe don't have a background in mushroom cultivation, just some of the basic needs you'll have to provide for when you design some kind of, setup to digest with fungi, common toxic waste. They need food. So typically the waste is their food. But sometimes if it's super toxic, it's really helpful to give them a little bit of easier to digest substrate. And so, for example, with the bike rag project, the rag itself was actually an easier to digest substrate. So a lot of clothing is made out of cotton, which is a dead plant. So in that instance, I actually didn't need to supplement with an easier to digest substrate for the mushroom to grow just fine. But there's other instances where it's helpful to add, a little bit of freshly spent coffee grounds, or a little bit of cardboard, or a little bit of some kind of dry straw or something. It just helps the fungus grow healthy and strong and resilient, and then have the time it needs to figure out how to digest that harder to digest stuff. The substrate always needs to be moist. I mean, this is why the meeting started with lamenting, the lack of mushrooms this year. There wasn't enough water. You all intuitively know, I'm sure, that for fungi to grow, they need water. So and something that isn't always understood is that a lot of fungi, not all, do need air. They need actually oxygen. And so what that means, I'll give a quick example, is so when I was doing this system, you can kind of see I actually have holes in my bucket, so that those holes provide for air exchange. When I scaled up to a much larger system, actually one of the main problems I had at first is there were these pockets of where there wasn't air. And so I actually had to devise a system to provide air through this entire space so that the mycelium could grow all the way through. And so that's why I always like to, in all my workshops and presentations, just mention the basic needs of fungi, because if one, you're going to have to design to meet those in the first place for your project to work. But whether that's for remediation or cultivation for food, or for materials for that matter, you're going to have to provide for their basic needs. And if something isn't working, I would encourage you to think back to, okay, wait, so maybe, okay, does it have enough food that it can eat? Is the substrate, is that food wet, moist enough? Is there enough moisture? Oh, is there enough air exchange? And so that's why you can see I would always pre-soak the rags before I put them in to meet those needs. So just to sort of kind of wrap up this model. So the basic sort of protocol or method that I developed is actually growing oyster mycelium on cardboard sheets. So again, cardboard that's already addressing a major, part of the waste stream. Oyster, because it's really easy to grow in a contained system. It grows great on on cardboard, and then you end up with these nice, like, flat planes of mycelium. Those are really good to work with. So then you'll need to collect your waste material, and you always will soak it before you inoculate. So I actually would always have a special kind of soaking bin, because of course there's a somewhat type of wastewater that's created through that soaking, but you can just use that to water the system. And then you'll basically add your soaked waste into the mycodigester, ensuring adequate air exchange. And so I played around with different methods, and it kind of depends on the type of waste and its surface area, like how small it is, how many edges for mycelium to hook onto. But I found that like for cigarette butts, it worked actually better, or it worked great just to actually add them on top of the growing mycelium, and it'll crawl up and you can just keep adding more soaked cigarette butts. With the rags, I found that layering worked well. So that, you know, I would layer the sheet of oyster on cardboard with a soaked rag and keep going. And then I'll say too that that didn't do the trick all the way. So I found that after that, it's sort of a primary decomposition that gets the process going, definitely reduces significantly, you know, the organic contaminants for the ones I tested. But I suggest then, you know, maybe you inoculate that with a secondary fungal decomposer. Maybe you compost it. Maybe you add it into a worm bin. When I did that, it took the toxic waste all the way from like this, you know, like a pile of, you know, cigarette butts or a pile of Teflon-based lubricant-soaked rags into soil. And so if that's your goal, to create non-toxic soil out of a previously toxic waste stream, then you're going to probably need to compost or do a secondary decomposition. And on my website, I have, on my blog, I have little handouts and walkthroughs for some of this stuff. But, you know, if you have an oil leak with your car, you can use this same method to deal with it. You know, there's further questions that I know I'd like to continue to test. Yeah, I hope that this just provided some ideas to kind of get started. I'll then close with the third, definitely not last, but the third potential for mycology to help address waste, and that is to actually replace toxic and environmental costly material, so that we don't even have styrofoam and other plastic packaging and stuff in the landfills, because they're made out of biodegradable mycelial materials. So Ecovetive is a leader in this space. You know, they're the ones who are making the like styrofoam replacement, the packaging replacement that I think IKEA now is using, and other bigger companies for their packaging. They're also making you know, leather, like mushroom leather and mushroom hides and other materials. But this is a really interesting space where there's a lot happening right now, a lot of exploration. I saw a study for some of you who may be gardeners, maybe grow food in other ways. You may know that peat moss is really a common part of a growing media mix, and but it's actually mined from peat bogs, and it's running out. We're running out of peat moss. So actually spent mushroom spawn from growing operations has been studied and found to be a pretty good replacement for peat moss. For those of you who have seen, you know, mycelium, once it's you know, dried up, it's quite fluffy. And so I'll just end. This is a new collaboration I'm working on with the textile artist Dominga Pazo Pavez. And so you saw, you know, textile waste is one of the top 10 components of the waste stream. You know, textiles contain toxic dyes and plastics and all sorts of other materials. And so we're working together on this project called Redress, working with fungi to transform textile waste into soil and also into objects. And so, wow, I somehow ended pretty much right on time. So really thank you for coming tonight and for listening. And, you know, I'm excited. I'm part of the, you know, I'm a member, so hopefully see you all around at different meets, but you know, I live locally and work on this stuff locally. So if you have, you know, if you want to reach out, please do. My email's here. I have a blog that I don't spend nearly enough time, you know, updating, but there are some handouts and some information on there, and I'm on Facebook and Instagram and everything as well. So yeah, please do let me know if you have any questions, and please do, you know, connect and reach out if you'd like to work together, or, you know, you have ideas. Thank you.
Thank you so much. That was great. You covered a lot of stuff. There's some really neat applications there. We'll go ahead and take some questions. If you want to, Rich, can they unmute? I can't remember. If you want to
Yes, people
Yep.
can unmute.
You can unmute as long as we're good. Go ahead. I'm not seeing one right there right now, except for lots of things like wow and great presentation and thank you. But I'll go ahead and ask a question because there were a couple of things that struck me. One is in an electronics waste pile, like the one you showed, where do fungi get enough water to like start breaking down what seems like really hard, difficult stuff to break down?
That's a great question, and I mean, I'm amazed too. I don't know. I mean, that pile looked to be outside, so you know, outdoors there'd be ambient moisture, you know, depending on the climate and you know, potential rainfall. But I will say that, you know, the focus here was more on waste, but I also work on remediation of, you know, land, more specifically too. And it's just, I'm always amazed at how, I mean, you may have heard there's, you know, there's fungi growing towards the radiation in, you know, melted down nuclear reactors at Chernobyl and things like that. There's fungi that just seem to really thrive in really harsh environments with little water, you know, and really high levels of toxicity. So yeah, I don't know how they do it. I mean,
Yeah.
I know how like some fungi, I mean, I know the mechanisms for mycorrhizae and certain fungi, how they survive without much water. You know, they actually create sticky substances that kind of grab onto whatever water there is and hold it near them. But I don't know for that particular example, it's pretty amazing.
Yeah, I didn't have I didn't know if you would have an answer.
Yeah.
I'm just curious if, but you might.
Yeah.
And, you know, I mean, for example, all those parts get handled when they're manufactured, but a lot of them are manufactured under clean conditions. So I wondered,
Right.
for example, if things like fingerprints were like the maybe
Ooh.
co-substrate or something like that.
Right. Yeah, that's good thinking.
Do we have any other, here's a question. I don't know if you can see it or I'll read it.
Sure.
From Elizabeth. Is it safe to use cardboard from a shipping box, example, Amazon boxes, as a substrate and then use the digested material in a food garden?
Yes, yeah, that's a great way to build soil, actually. And so the only exception would be boxes that have glossy colored ink. But any sort of regular, I mean, I'll say too, waxed boxes, those are just harder for the mycelium to eat. The wax kind of protects the paper, the cardboard. But yeah, so but a regular cardboard box without glossy colored ink is totally fine, even if there's you know, writing on it or something. Dyes are soy based, and the glue that holds boxes together are actually lignin based. So that stuff that that those mushrooms love to eat anyways. And so what's nice about growing on cardboard is, man, those, oyster mushroom, King Stropharia, its Latin name is Stropharia rugoso annulata. There's a number that there's nothing better on earth than them at eating through cardboard. And it's just like I said, it's a great way well, you could grow some mushrooms to eat. And then you can put all of that material into your garden. It'll just create amazing soil. Worms love mycelium, so they'll come up, and before you know it, you'll have amazing soil in your food garden.
Great, thank you. I think that might have been a very widespread question. I see another question from David Roman Daniels, and it is, what is the name of the peat most substitute? David, I'll have to try and interpret that. Do you mean the best mushroom substitute?
Oh, so actually, that's so cool that you're asking about that. I mean, I actually haven't found it on the commercial market yet. This study, when did it come out? Okay, this study came out 8 years ago. So, I mean, if it's not on the market, maybe you'll be the one to to get it out there, because it seems, sounds like you might be a food grower. But you'll know, this is a major problem. A lot of food growers are really concerned about using, it's just not as available anymore because it's sort of non-renewable. So I think this is a great idea. a lot of times folks who run commercial mushroom operations, the spent substrate is a waste stream for them. And I always think, hey, no, you can, it's a soil amendment. It's useful in remediation. You dry it out and fluff it up, shred it, and it's a peat moss substitute. So there's really sort of so many options. I haven't seen it though I haven't, seen it in a store where I could buy it, but I think someone should do that for sure. Oh, so Hope found a manufacturer and put the link in the chat. Thank you so much, Hope.
Okay, and I also see a question from Gustavo Alvarez. And the question is, can mycomaterials be reused?
So in terms of like packaging and things like that, I don't know, because it it will break down. It's truly biodegradable. So I'm not sure how many times certain myco materials can be reused. It really depends. So if it's intended to be a single-use item, I think it breaks down. It's used once and then it breaks down basically and becomes soil. But there are other myco materials like bricks, myco bricks, leather, that are, and so many other that are intended, they're designed to be durable materials. So in those, it depends on how they've been designed, whether they've been designed to be sort of a one-time use replacement for packaging or other one-time use items, but at least biodegradable and grown on waste, or they've been designed to be a durable and long-lasting material that has use in like, building or clothing or or things like that.
I see a question from Aaron Thompson. He wanted to know if you were recently in Mycotopia.
I was, yeah. Doug Bedard's.
Tell us about that.
Oh yeah, that was cool. Doug visited. Oops. Oh, I'm gonna stop my share. Doug actually visited me at UC Riverside, and in that book, the focus is more on my research into mycorrhizal fungi. And yeah, so it's sort of a snapshot into what I'm working on now at UC Riverside. Yeah, that's a really cool book. I've been,
Where would they find that? And don't say
it's
Amazon.
through, so I believe it was Chelsea Green was the publisher.
Okay.
I have it somewhere. Yeah, so you can order it online through Chelsea Green, the publisher directly, and other sources. It's a really cool book. It's a little different because it's looking at sort of the culture around mycology in North America. So yeah, and sort of citizen science and mycology. So it's kind of neat. Oh, thank you. Wow, y'all are awesome. Okay, so Gustavo actually put a link in the chat to where you can purchase that
Excellent.
book.
Thank you, Gustavo. And by the way, I'm the one earlier who mentioned using Amazon for some of my stuff. Sorry, everyone, but that is a source of place. So I see something from Amy Hongio, and if you use mushrooms to break down cardboard and then add this to your garden, will more mushrooms grow in my garden?
Probably, yeah. Yeah, so, and that's good. So sometimes when, when mushrooms have stopped fruiting, as soon as you put it outside in nature where it can access more moisture, more food, really the reason it stops is because it's out of food. And so once you put it in the garden, it probably will grow again, particularly the Stropharia I mentioned. It's an amazing, common name, garden giant. It's a great companion to the garden. They're decomposers, so they won't harm your living plants, but they'll build soil out of what they're growing on, and then you might get some bonus crops that keep, popping up.
Are there any questions I missed? If some of them came in earlier, I might have missed them. If someone wants to speak up, or if someone else wants to say something they saw in the chat and point it out. I had one last question I wanted to ask, which was in that little rain garden thing you described, do you have an idea for the mechanism of reducing coliform bacteria?
Yeah. So, well, okay. There's a number of different species that are all working together. So there's different mechanisms for each, but basically the plants, so remember there's native plants, and then there's these different decomposer fungi, and the plants have their mycorrhizal partner. Every one of those species actually needs nitrogen and phosphorus for its life processes. So you might know this, you might have seen, like, sometimes even oyster mushrooms they'll trap living nematodes to get the nitrogen they need. So all of these need nitrogen, and actually nitrate and phosphate are the common excess nutrients mentioned that run off from agriculture, and they cause problems in watersheds. So the mechanism of those excess nutrients is just that they get sucked up and used by all of those plants and fungi.
Okay.
For the fecal coliforms, it's really the decomposer fungi, particularly again that Stropharia, but also the oysters that go to work. And the reason, so basically they are able to inactivate fecal coliforms. And the reason they can do that is because they're actually, they're, competitors and predators for them, right? These are fungi that live in soil environments where there's a bajillion, other microbes and things. So they have to learn to survive. They have to be able to deal with, fecal coliforms are also a pathogen for them. So they're able to inactivate them, and they do that so that they can survive. But that's also why they're just pretty effective filters if installed the right way. But the combination of the plants is really important there too.
Do you see that last one there from
Sure.
David Nussbaum? Any
Oh,
thoughts on licensing approach of companies like Ecotative? Should others be allowed to utilize similar approaches to create similar products?
I'm not super familiar with their licensing approach, but I mean, I guess I could think about, but I guess for me, I'm way into, I mean, obviously I'm an educator, and I really share what I'm working on, and encourage others to try things as well. So yeah, I think I'm very very encouraging of sort of open source, especially for things that really benefit benefit the world. But I know that that's a bit different than how companies, some of the bigger companies do it. But I think, yeah, I'll say I still think there's a lot of space for people, for mycologists to make contributions to this kind of whole area of applied mycology. And so even though there are some companies that are kind of claiming some territory and with certain methods, there's still so much to figure out, and still so much. And there's a whole, as much as there's kind of the biotech kind of side of things, or movement, I guess, there's also equally, at least, sort of more citizen science, community science, open source kind of approach going on. So I'm encouraged by that too. Yeah, I don't know if that answers your question. I'm not super familiar with their specific licensing approach, but I do know that there's, yeah, I somewhat, yeah, I still would encourage people to explore mycomaterials if you feel called to do it. Yeah.
Cool. Any other questions for tonight's speaker? Well, thank you so much for coming tonight. I'd like to say that I think this concludes it does conclude the regular season for the Los Angeles Mycological Society, which has been conducted entirely online. And so we have one last meeting, which is we have a special meeting coming up tomorrow. Just remind people of that. If they'd like to come on and vote, please do. It's open to all members to vote. And then the annual meeting will be coming up. It's usually a potluck, and that's not going to happen. I don't know if there's not an agenda for that meeting yet. If there is, it will be sent out by a LAMS email, and you'll have a chance to respond and or attend virtually, that is. I'm saying one or two more things here besides the the exclamations. I'm just seeing Gustavo, or you were in Riverside, please let's connect. Kate, same, Gustavo, and so you're getting a few there. This is recorded, and so all the chat will be available to you to go back through in case I missed anything. So thanks again, everyone. Thanks for coming tonight. And thanks for this excellent presentation. It's really nice to have, and we really appreciate it. And we hope, since you're a LAMS member, you can come in person someday soon.
Definitely, I hope so too.
Yeah. Okay, great. Well, thanks again, everyone. I'm gonna say so long, and really appreciated it and enjoyed it. So enjoy the summer if we don't see you at the special meeting or the annual meeting, and hope for rain in the fall. Take care.